We will now look at how to set up and connect a microplate respirometry system for use in our MicroResp software. Before setting up the required hardware, we recommend downloading and installing the latest version of MicroResp directly from our website. Start by inserting the LollyGo license dongle in a USB port on your PC to unlock the software, as MicroResp will run in demo mode, indicated up here, if the license dongle is not inserted. Running MicroResp in demo mode will show virtual hardware with simulated data and will not allow you to connect any real hardware. But the analysis functionality runs without limitations, even in demo mode, so you can analyze your data files on any computer running just the demo version. No need for a license dongle here. Now connect the microplate reader power supply to the splitter, and connect the splitter to your PC via the USB cable. Connect the reader to the splitter. If you have any additional readers, simply daisy chain these to the first reader using the short yellow reader cables. Now connect the power supply to a grounded wall outlet. The microplate reader is now ready to be used in MicroResp. It's important to maintain the temperature of the glass microplate, as the optical oxygen sensor spots inside each well are very temperature sensitive. So even small variations in temperature will affect the oxygen readings. Keeping the temperature stable, whether you are measuring in water or in gas or air, can be done using our optional flow-through water bath. Simply place the water bath on top of the microplate reader and place the sealed glass plate inside the water bath. Now use a refrigerated bath to run thermostated water through the bath. Note that you do not have to fill the entire water bath with water, as the water just has to cover the glass plate. If you're using the optional silicone pad and compression block, cover that too with water. Alternatively, you can use an incubator to maintain the temperature. If using an incubator, place the included white plastic guide on top of the reader, the glass plate into the guide, and then keep only the reader, guide, and glass plate in the incubator. Note that the long yellow reader cable comes with a flat black part, so that you can keep the incubator door closed. Finally, the entire microplate system can be placed in a climate-controlled room. We recommend leaving the setup at the experimental temperature for around 60 minutes prior to an experiment to obtain a stable temperature before starting measurements. For aquatic measurements on immobile organisms like eggs or embryos, we recommend placing the reader and the plate on an orbital shaker table to somewhat agitate the water, as this will reduce oxygen stratification inside the wells. Cleaning the glass plate can be done using ethanol, bleach, or mild detergent, and then rinse with demi-water. Now allow the plate to dry. If using ethanol to sterilize the wells, the oxygen sensor spots must dry out thoroughly for at least 2 days at 50 to 60 degrees Celsius in an oven to ensure that all alcohol residues have left the sensor dye matrix. We recommend performing a manual 2-point calibration after sterilizing. The optical oxygen sensor spots used in the microplate system can be sensitive to light sources, especially UV light. So we recommend avoiding exposure of the reader and glass plate to strong sources of light during measurements, as this can interfere with the oxygen readings. It's also why we advise storing the cleaned glass plate in the non-translucent black plastic bag between trials to prevent exposing the sensor spots to light and thus bleaching the sensor dye, causing signal drift. And that's it on how to set up the microplate respirometry system for use in MicroResp. If you have any questions about setting up the systems or about other aspects of the system or software, you are welcome to contact us on our website.